Hi, welcome to Cell Bio News. My name is Fabio, and today I want to talk to you about malaria. Now, malaria is a nasty little bugger. It kills a lot of people. And it used to be that you could treat malaria with a drug known as chloroquine, or CQ, in which was, I quote, cheap, safe, and effective. Now, the malaria parasite eventually developed a resistance to this drug, and scientists didn't know why or how until recently, when Rowena E. Martin and friends published an article in Science magazine called Chloroquine Transport via the Malaria Parasite's Chloroquine-Resistant Transporter. Now let me tell you a little bit about how chloroquine works. Chloroquine would enter the vacuole, the digestive vacuole, of the malaria parasite, which is a lysosomal organelle, where it would accumulate. Because CQ can enter the lysosomal cell because it is a weak base, and the lysosomal organelle is very acidic inside. And once it entered via simple diffusion, it would gain 2 protons and become CH2+, where it would then be stuck and not be able to simply diffuse out again. Now, it is inside the vacuole that CQ was thought to exert its anti-malarial properties, and it would interfere with the detoxification of heme, which is a byproduct of the hemoglobin, thus killing the malaria parasites. Now, through this publication, they found out that the resistance was mediated by the Plasmodium falciparum chloroquine resistant transporter, or PFCRT. Now, this protein had a key mutation, which was the key resistant conferring mutation in the PFCRT, which was a replacement of the lysine, K, with a threonine, T, at position 76. Thus it's called a K76T mutation. Now what this did was it removed a H+, or a positive proton, from the ligand binding site, which is what prevented the CQH2+ from exiting. Once this was removed, the PFCRT could transport the CQ out of the system, thus allowing it to not accumulate, and so the heme wasn't able to be interfered with, and so the malaria was resistant to the drug. Now this is a very good finding, as they can now figure out how to make new drugs that can effectively reverse this mutation, or at least prevent it from interfering with the CQ accumulation. They tested a drug called verapamil, which actually does reverse the mutation and adds another H+ into the ligand binding site, which allows again the accumulation of the CQH and causes the malaria parasite to die. And so it's possible that others can actually, they say, very easily block this binding site and prevent this mutation, or at least reverse it, which is a good thing. So yeah, yay science!